أعوذ بالله من الشيطان اللعين الرجيم بسم الله الرحمن الرحيم اللهم صل على محمد وآل محمد وعجل فرجهم. If we take overview of molecular diagnostic techniques and instrumentation, then first of all we have to discuss reverse transcription polymerase chain reaction. This is also abbreviated as RT-PCR, and it uses RNA as a starting material for in vitro nucleic acid amplification. The discovery of retroviral reverse transcriptase in early 1970s ultimately made RT-PCR possible. Reverse transcriptase is an RNA-dependent DNA polymerase. It is DNA polymerase, but it is RNA-dependent DNA polymerase, and it catalyzes DNA synthesis. And using RNA as template, the end product is known as complementary DNA or cDNA. cDNA is not subject to RNA's degradation, so making it more stable than RNA. So if question is raised that what is the benefit of making of cDNA, then very simple answer is that cDNA is not subjected to RNA's degradation. It is not degraded by RNAs, and it is more stable than RNA. In RT-PCR, the starting RNA is subsequently degraded, double-stranded DNA is produced, and PCR amplification proceeds in the usual manner. Very simple. Again, we have to focus on this point that in RT-PCR, what happens is that starting RNA is subsequently degraded, number one. Number two, double-stranded DNA is produced, and then PCR amplification process proceeds in usual manner. So it is the combination of two main techniques, number one reverse transcription, and number two usual PCR, usual amplification of the DNA. RNA extraction kits for both manual and automated RNA purification exist, and when combined with RT-PCR make RNA analysis in clinical laboratory virtually as rapid and equally sensitive as PCR based DNA amplification. So RT-PCR is commonly used in the diagnosis and quantification of RNA viruses, and for quantification of RNA virus infections like human immunodeficiency virus can be diagnosed by RT-PCR, hepatitis can be diagnosed, and in animals, foot and mouth disease can be diagnosed, and other viral diseases, they can also be diagnosed by using this RT-PCR. And the analysis of messenger RNA transcripts such as those produced by translocations commonly associated with Hodgkin's lymphomas, leukemias, and sarcomas. So gene expression profiling is likely to have a major impact on molecular diagnostics in the coming years, and will depend on RNA analysis using RT-PCR and possibly high density arrays. So main benefit of RT-PCR is the identification and quantification of RNA virus infections related with human diseases and animal diseases, and yes, of course, in plant diseases as well. and the analysis of messenger RNA transcripts, such as those produced by translocations. So this was a little bit about RT-PCR. Thank you very much. Jazakallah khaira. Allahumma salli ala Muhammadin wa ali Muhammad wa gajjil farajahum.